Hello everyone, this is a review on radiobiology. We're going to talk about 5 topics from Bouchon's textbook in this recording. The first one is human biology from chapter 29 of his textbook. The next one is fundamental principles of radiobiology. And then we're going to talk about 2 chapters combined on molecular and cellular effects of irradiation, and end with deterministic effects and then stochastic effects of radiation. Let's begin with Bouchon's chapter 29 on human biology. As far as human radiation responses are concerned, the effect of X-rays on humans is the result of ionization at the atomic level. The result is absorption. Absorption can be due to the Compton or photoelectric interactions, Compton being partial absorption and photoelectric being total absorption. The end result is either a short-term or long-term type of damage or effect on the body or the person themselves. Human responses can either be early or late. Early effect of radiation versus late effects are both radiation responses, but they're not occurring within a certain time frame. So, early effects of radiation are radiation responses that occur within days or weeks after radiation exposure, while a late effect is radiation response that is not observed for months or years. An example of an early effect is erythema, and an example of a late effect would be cancer or leukemia. Radiobiology is a study of the effects of ionizing radiation on biologic tissue. And a caution to keep in mind is that even small doses of X-rays are harmful. We'll talk about this again when we discuss the linear non-threshold model, in that any dose of radiation can cause an effect. Let's examine the composition of the body. In terms of the atomic composition, notice that our majority of our bodies comprised of hydrogen at 60%. 25.7% of our bodies are comprised of oxygen, which is the reason why we have H2O is our most common molecule at 80%. Notice that the atomic composition then decreases to carbon being the third most common atom in the body, while on the molecular composition, 15% of us are comprised of proteins, then lipids, 1% carbohydrates, 1% nucleic acids, and 1% other. I'd like to take a moment just to talk about the nucleic acids. So we have 2 types of nucleic acids. They are DNA and RNA. When you consider that 1% are nucleic acids, that means that less than 1% is DNA. There are 2 categories of molecules in the body. They are organic and inorganic. The difference between the two, the major differences, are that organic molecules are macromolecules. That means that they're very large, hence the word macro. And they consist of sometimes hundreds of thousands of atoms. Inorganic molecules, on the other hand, do not contain carbon, and they're going to be much smaller. As far as the 4 types of macromolecules, there are proteins, lipids, carbohydrates, and nucleic acids. There are 2 types of nucleic acids, and they are DNA and RNA. When we talk about proteins, they're comprised of carbon, hydrogen, nitrogen, oxygen, and other things. I call them stuff. So I tend to think of proteins as schnoz, CHONOS. Carbon, hydrogen, oxygen, nitrogen, oxygen, and other stuff. Schnoz. And then lipids and carbohydrates are both comprised of the exact same types of atoms. Those are carbon atoms, hydrogen, and oxygen atoms, CHO. So they're both CHO. The difference between lipids and carbohydrates is that they're just structurally different as far as the amount of ratio of carbon to hydrogen to oxygen. The reason why carbohydrates or saccharides are called carbohydrates is because they were first thought to be water carbon. So their structure is CH2O, which is water and carbon. Lipids are different. The difference between lipids and carbohydrates as far as what they do for us in the body is that lipids are more kind of like looking more like glycerol and fatty acids. In fact, there's the lipid molecule is comprised of one molecule of glycerol and 3 molecules of fatty acids. The lipids are going to be present in all tissues, and they're going to be found in the structural components of cell membranes. Lipids are going to be oftentimes serving as a thermal insulator from the environment. They also serve as fuel for the body. If you think about it, they're going to be a long-term energy store versus saccharides or carbohydrates are going to be short-term energy stores for the body. Finally, we have nucleic acids, and those are the DNA and the RNA that we talked about previously. And your author goes into a lot of detail talking about DNA and RNA and the structural differences even between them. Keep in mind, just as a review from biology class, is that DNA has a double helix. It has adenine, thymine, cytosine, and guanine, where RNA only has a single spiral and uracil replaces thymine. And then finally, the sugars are different. When you look at DNA, the name of it is deoxyribonucleic acid, so its sugar is deoxyribose. And with RNA, the sugar is ribose. RNA stands for ribonucleic acid. So they're named after the type of sugars they have. With regard to the inorganic molecules, they are much smaller. They're not going to have carbon at all. And in fact, water is just H2O. When we consider how important that is to us, it's extremely important. I mean, it's 80% of our body is comprised of water. Where mineral salts are highly important to us because if you think about the electrolyte balance, or you may have heard people come into the emergency room and their electrolyte balances are off, that's referring to mineral salts. And so those are going to work with nerve conduction and so forth within the body. So it's highly important that we have water and mineral salts too. Cells are the basic units of all living matter and essential for life. You've had this previously in biology classes and anatomy classes, so I won't take a lot of time on this either, but it's important for us to review a couple things about the cell. When we look at the cell, we have to keep in mind that it moves, it grows, and that's something we're going to be emphasizing in this chapter 2 is about the growth. We'll talk about it reacting, protecting itself, regulating life processes, and reproducing. There are 2 major structures of the cell, and that's the cytoplasm, in which all metabolic functions are carried out, and it has or contains all the cytoplasmic organelles and all cellular components except for DNA. So it'll have things such as the endoplasmic reticulum, the ribosomes, the mitochondria, etc. Where the nucleus, on the other hand, is the center or the headquarters of the cell. It's like the brain of the cell. And when you consider the importance of that nucleus, that's where DNA is going to be hanging out. It's the only location in the body that you're going to find DNA. So it's a very important thing too. It's not just that you have just DNA there, you also have some RNA and water, but it's important to note that the cytoplasm has everything else. Cytoplasm also has water and RNA too. With cell structure, just to bring that up again, the nucleus, just further emphasizing, is going to be a round mass of protoplasm. It has a double-layered membrane. In this particular diagram, it shows it being called a nuclear envelope, but many times it's called a nuclear membrane. It's a double-layered membrane that allows the structures in the nucleus to exchange information with the cytoplasm and to move things in and out as need be. It contains DNA, RNA, protein, and water. But keep in mind the cytoplasm also contains RNA and protein and water. The nucleus is the only location that you'll find DNA. The function of the nucleus is that it's going to control cell division and biochemical reactions within the cell, and specifically DNA is in charge of protein synthesis and cell division, so it's highly important. Within the nucleus, we have chromosomes, and these are tiny rod-shaped bodies that are composed of DNA. In humans, there are 46 chromosomes, otherwise known as 23 pairs. And we can see in the diagram on the right-hand side, where if you take a cell with a nucleus, and then there's chromosomes within the nucleus, you blow that one chromosome up after its S phase, and then we unravel that further. It's so neat how we have so much DNA in our bodies. It's incredible the amount that you've got. I want to talk about a few of the cytoplasmic organelles that are inside the cell, within the cytoplasm itself. Here's the mitochondria. They're bean-shaped structures. They are digested, and the macromolecules are digested in it to produce energy for the cell. And so if you think about beans producing energy, that's one way to think about that kind of thought process. Secondly, we have the lysosomes. They're little pea-shaped structures, and they are going to contain enzymes that are capable of digesting cellular fragments, and sometimes the cell itself. It's going to help control intracellular contaminants, get rid of things like a garbage disposal system. Another important structure that we're going to have are the ribosomes. They are little dot-like structures, and they're the site of protein synthesis, and therefore essential to normal cellular function. Keep in mind that your ribosomes can be scattered throughout the cytoplasm or attached to the endoplasmic reticulum or reside on the endoplasmic reticulum. The endoplasmic reticulum, or ER, is kind of like a bridge within the cell structure, and it's going to be a channel, a set of channels or series of channels allow the nucleus to communicate with the cytoplasm. There are two main categories of cells in the human body, somatic and genetic. The term somato in medical terminology means body. So somatic cells are cells of the body. On the other hand, genetic cells are reproductive or genetic cells. The type of division under which somatic cells go is mitosis. And the type of division under which genetic cells undergo is meiosis, otherwise called meiosis, or pronounced meiosis by some individuals too. When we look at the difference between these two types of cells, an example would be a sperm cell or an egg for genetic cells, and all other cells in the body would be considered somatic. So an example of a somatic cell would be a red blood cell, a white blood cell, nerve cell, skin cell, any type of cell you can imagine, a muscle cell, anything like that. So somatic cells are anything in the body, genetic cells are reproductive or genetic. The next thing to take a look at that's very important is the cell cycle, otherwise called the cellular life cycle. The thing that Bouchon that brings up in this chapter is that cell biologists and geneticists view the cell cycle differently. Each cycle is going to have a stage of growth, development, and division. We're going to, when you're looking at talking about geneticists, they're only going to consider two phases of the cell, mitosis and interphase. We're going to break it down a little bit more in terms of four parts to it, in terms of biologists. So when you're looking at the cell, it undergoes a period of interphase in which there is G1, S, and G2. G1 is referred to as the pre-DNA synthesis phase. So it's a phase during which the DNA is getting ready to divide. It's a gap phase. And then you have the S phase, which is called the DNA synthesis phase, in which we're going to duplicate DNA into identical DNA molecules from what we originally had. And then we have G2, which is another gap phase. It's going to be another phase during which there's more regrowth and formation happening after the S phase. So it's called post-DNA synthesis phase. And finally, M is mitosis. So repeating that again, G1 is pre-DNA synthesis phase, or gap. S is a DNA synthesis phase in which we duplicate DNA. We get double the amount we originally had. G2 is the post-DNA synthesis phase, or gap. And then M is mitosis. The stages of mitosis are prophase, metaphase, anaphase, and telophase in order. They're PMAT. The entire cycle has to occur in this order in order to have mitosis occur. And so it's very important that we consider the whole cycle. Many times the interphase is considered just a resting phase during which nothing happens, but that is not the case. There's actually quite a bit happening during interphase. So interphase, just to repeat that, is where we're going to have a second set of chromosomes being produced by the cell, and we're going to store energy. So it might look like it's dormant and just resting, but it is not. The G1 phase, just to review, is called the pre-DNA synthesis phase. During this phase, specifically, RNA synthesis occurs. We're going to get extra RNA growth happening. It lasts from just a few minutes to several hours. G1 is also referred to as a gap, but it's pre-DNA synthesis. The S phase is the DNA synthesis phase. It's a very long process, a long time frame as far as a part of this cycle. The chromosomes change from having 2 chromatids attached to the centromere to having 4. This can last a maximum of 15 hours. Again, very slow, long, laborious process. G2 is referred to as a post-DNA synthesis phase or second gap phase, and this is the phase during which RNA and protein synthesis phase occurs. It can last from 1 to 5 hours. The difference in length of time for these different phases varies upon the different type of cell to which we're referring. So for example, lymphocyte, the length of time for a lymphocyte to go through these phases would take much less than would a nerve cell. In the S phase, which was called the DNA synthesis phase in the previous slide, or the one before that was the green letter, a green area on our chart, is where we have 2 chromatids attached to a centromere. So here's one at the top and one at the bottom, and here's the centromere in between. And during that phase, we're going to replicate the DNA or duplicate it to the point where it has 4 chromatids now attached to the centromere. So there's a lot of things happening during that phase. Since it takes so long, this is the least radiosensitive phase, otherwise called the most radio-resistant phase of the entire cell cycle. During the M phase, otherwise known as mitosis, in the cellular life cycle, we have division of somatic cells, in which there's one cell with a diploid or 2n number of chromosomes that divides to form 2 cells with a diploid number. So we're getting 1 cell to start with, and we're ending up with 2. When you're looking at this, it's like having a daughter cell, 2 daughter cells that are going to be equal to have exactly the same information as the parent did. And we'll put this in a diagram in just a moment. So you have 1 parent cell and 2 daughter cells. So just to go back to maybe, I remember learning this in the 8th grade, so it's been a while, is a diploid number is a 2n number, and n means the number 23. These are referring to somatic cells. If you think back to the number of chromosomes you have in your body, 2 times 23 is 46. So these are the cells of the body that are dividing, that we're going to go through the process of mitosis. Your 2 daughter cells have the exact same information, genetic material as the parent cell did, and during that cell cycle, the cells are most sensitive during this whole process during mitosis, because that's when the action's occurring. That's when the cell is the most active, is during this phase. And it's the most resistant when the process is slow. As we talked in the previous slide, it takes up to 15 hours to go through an S or DNA synthesis phase. Prophase is the first stage of mitosis. You can think of pro as pre, first thing happening. During this phase, the nucleus swells and the DNA becomes more prominent and begins to take structural form. It uncoils, I guess you could say would be a good, or coils, The nuclear membrane disappears, and the centrioles contained within the centrosomes move towards opposite sides of the cell and organize what are called the spindle fibers, across the equator. Metaphase can be thought of as being middle, and that's where the chromosomes are going to line up across the center or the equator of the cell. So I call it the metaphase or middle stage. This is also a good time during which the centromere of each pair of chromosomes is attached to a spindle fiber. Notice that the DNA have 4 chromatids attached to a centromere because we've already gone through the S phase. So each one of those centromeres is attached to a spindle fiber at this point, and we'll have a total of 46 across there doing this across the equator. So this is just a small version of what we have, of course, macroscopically considering we normally can't see this at this level. And we can best analyze damage under a microscope during this phase. So chromosome damage can be analyzed during metaphase under a microscope. That's your 2 blanks there, during metaphase under a microscope. Anaphase is a phase during which where this centromeres split. Now we only have 2 chromatids attached to a centromere. They've split and they're going to opposite poles. So ana means away. Spindle fibers contract and pull the chromosomes one set toward each pole of the cell. So they're away from the equator, away from the center. Telophase is the last phase, and telo or tele, T-L-E-O or T-L-E-T-E-L-E stands for far, F-A-R. And so the chromosomes reach the poles of the cell, which means they're going far away, and DNA uncoils. The nuclear membrane reforms around each set of chromosomes, so it's going to be contained, each are going to be contained within the nucleus again, a nucleus, and the cytoplasm is then divided into 2 parts. At this point, the spindle fibers are going to be going back within the centrosomes. We're ready to head back to interphase at this point. This is a picture from your textbook, specifically from Bouchon's textbook. And it's on page 478. What we have here is we've got A is where the stage of interphase is being shown. They went ahead and showed you, I guess you can say like prophase is happening as well, prophase or interphase with prophase. Metaphase is that middle stage. They went ahead and showed you where the centromeres are attached to the centromeres of the chromosomes are attached to the spindle fibers in the equator of the cell, and that's during metaphase. Here's anaphase with C, D, and E, kind of like showing the phases of anaphase and telophase kind of happening. Now, what happens is this is originally at A was considered a parent cell. So this is a parent cell in the beginning, and at the very end, they're showing 2 cells at the very end at letter F that are considered daughter cells. So in the beginning, you have a parent cell, could be one nerve cell with 46 chromosomes. At the end, you have 46 chromosomes in each one of these cells, and those are both identical to each other and identical to the parent cell, but it again has 46 chromosomes. Has a diploid number. One cell, the diploid, forms 2 cells with a diploid number of chromosomes. Another type of division we have in the body is meiosis, and some individuals, like I said earlier, pronounce it meiosis. Division of different types of cells, so we're going to talk about this one now. So we have a division of genetic or reproductive cells. We oftentimes call them germ cells. The type of division we call this is reduction division. There are 2 types of reduction division. Since there are 2 types of reproductive cells in the body, there'll be 2 types of reduction division. The first type is spermatogenesis, which is the production of sperm, and oogenesis is the production of eggs. In the process of spermatogenesis, we have one cell with a diploid number, or 2N, and I put a little caption up at the top of the page that a diploid number, to remind you, is a 2N number, where N stands for 23, where we have 46 total chromosomes, because 2 times 23 would be 46. And it forms 4 cells with a haploid number. So there's haploid means N, and otherwise known as just 1 times itself, or just N, is 23 chromosomes. And so in the process of spermatogenesis, we have some one cell with a diploid number that divides 4 to become or form 4 cells with a haploid number. In oogenesis, we're producing eggs, unless there's a process of where there's a possibility of twins or triplets or quadruplets or sextuplets, etc. We're looking at one cell with a diploid number of chromosomes that forms normally one cell with a haploid number. If you have more than one cell, and you end up having twins or something. So taking a look at this from the standpoint of a picture, that on the top part, this comes from a different textbook, just shows one cell in the process of spermatogenesis called a spermatogonium. And a spermatogonium is the first stage. It's called the immature stage or first stage of a production of sperm. And it goes through the various stages that we go through with regard to going to duplicating the S phase, and then we get to going through the S phase, and we go through metaphase and the whole telophase and everything. We get to 2 cells with a diploid number at this point, and they split again to each one being a haploid number. So each one of these cells at this point has a haploid number of chromosomes, haploid being 23. At this point, they're called spermatids. They haven't fully developed, they don't have their flagellum, that kind of thing. In the final stage is called spermatozoa, where the sperm cells are fully formed, and they have now formed the correct figure of a sperm cell, plus they have the flagellum, which allows them to move. On the other hand, oogenesis is a process in which we have an oogonium, just the name of an immature egg, that's going to go through the same processes we have with spermatogenesis, except we get to the point of the end phase where we only have 23 chromosomes. We have one ootid, and the rest of them are called polar bodies. They have no function in the body and are just absorbed and discarded. Therefore, we have one ootid, and when it becomes fertilized by a sperm cell, it would be called a fertilized ovum, and therefore it would be the two would join together, each one having 23 chromosomes to make a person. So it only takes one sperm cell to fertilize an egg. Now, as far as tissues and organs are concerned, when we go from cells and we have collections of similar cells, they form tissues. And from that point, if we take cells that are precisely bound together, they can form organs. And the tissues and organs of the body serve as discrete units with specific functional responsibilities. So some tissues and organs combine to form an overall integrated organization, such as an organ system. So when you consider something like the nervous system or the reproductive system or endocrine system, respiratory, digestive, any of these systems, if there's damage to a certain system of the body, it can be due to a result of radiation injury to the cells of that system. For example, if you damage a nerve cell with radiation, you're could therefore impact the nervous system. There are 2 types of cells in the body, and they're identified by their rate of proliferation and state of development. And this excludes the somatic and genetic discussion earlier. This is just a different way to describe them. We look at these types of cells. We're looking at immature or mature cells. Immature cells are those that retain the ability to divide and divide rapidly. These cells are generally more sensitive to radiation than mature cells, otherwise known as being more radiosensitive. An example of an immature cell is in the process of spermatogenesis in the beginning stage, in which the cell was called a spermatogonium, the very first stage that we had is spermatogenesis. Another example, in the very first stage of oogenesis, we referred to the cell as an oogonium. And at the very first stage, those cells are, in that process, they are called immature cells. They are at that point dividing rapidly. The mature cells, on the other hand, no longer divide. So just we get to the point of being a spermatozoa, which is a sperm cell, there's no more division to go. So it cannot divide anymore. Or divides at a slower rate, which we'll give an example of in just a moment, than in immature cells, and they're less radiosensitive. They're also known as being more radioresistant. So let's take a look at the other names for these two types of cells too. So the immature cells are also called precursor cells and undifferentiated and stem cells, while mature cells are also called differentiated and end cells. Those are just other names for the same types of cells. An example here in your textbook on page 480 is that Bouchon states that stem cells are more sensitive to radiation than mature cells. So what it's saying is here's what stem cells up here, they're immature cells, they're going to be more sensitive than mature cells. This is a very important slide, and it's in your textbook as well. And what it is just showing the response to radiation as it relates to being the specific type of cell it is in the body. So the most sensitive cells in the human body are lymphocytes. They're the most sensitive, and this is in a continuum from being most to least sensitive. Notice that many people think back to, when you first started clinical, your first thought is gonads, I have to shield. That's not the most sensitive part of a person's body. So when you think about shielding yourself, you're more concerned about your blood cells, and specifically those related to your immune system. The second most sensitive cells in the body are the spermatogonia, and that's plural for spermatogonium. That's the first stage in spermatogenesis. Then erythroblasts are embryonic red blood cells. They're the immature red blood cells before they form an actual red blood cell. Intestinal crypt cells are located in the digestive system, specifically in the small intestine. Endothelial cells are in the lining of tissues. Osteoblasts are embryonic bone cells. Spermatids are the sperm cells right before they become spermatozoa, but they don't have the flagellum yet. They're still forming. They're still working in that production process. So it's the stage right before they are spermatozoa. Fibroblasts, they're embryonic fibrous cells. Muscle cells are myocytes. And then finally, nerve cells are the least sensitive cells in the human body. This is important to know from highest to lowest sensitivity. In terms of organs and sensitivity, they're determined by 3 factors. First is the function of the organ in the human body. Secondly is the rate at which cells mature in the organ. And third, just the inherent radiosensitivity of the cell type. So for example, your hemopoietic system, otherwise called hematologic system, is going to be where and specifically your spleen as one organ, will be the location in which you produce, and it's not the only one, but one of them, location for producing lymphocytes. So it's the function of that organ in the body is to do that, and they are going to mature very quickly. It takes less than 12 hours for a lymphocyte to mature, or it takes a lot longer for a nerve cell. So it's the inherent sensitivity of that cell type. And then lymphocytes are much more sensitive just by function of what they do. It takes a lot longer for nerve cells to grow back if they're dead than if you have damage to a blood cell, specifically a lymphocyte. So in terms of cell and organ system radiosensitivity, if we consider that lymphocytes are the most sensitive cells and they have the highest radiosensitivity, then the hematologic system, which is your blood system, would be the most sensitive organ system in your body. Then if you look at intestinal crypt cells as being a medium or less radiosensitive than blood and reproductive cells, some reproductive cells, then you'll find that the gastrointestinal system is kind of like a middle or intermediate system in the body as far as sensitivity. And the least sensitive cell in the body is the nerve cell, and so therefore the least sensitive system in your body is also going to be then the central nervous system. So that relationship is comparable between the cell and the organ system. Somatic and genetic damage are referring to damage at the cellular level, which led to both somatic and genetic damage in the individual as a whole. So thinking back for just a moment to somatic cells. Somatic cells are cells of the body. If there's somatic cells or cells of the body, such as a nerve cell, that's going to result to damage to the nerve itself, which is called somatic damage. So it's damage to that person. On the other hand, genetic damage is due to damage to germ cells or genetic cells, reproductive cells, to eggs or sperm. That's going to be impacting not the person, but to their future generations. So when you're looking at genetic damage, you're looking at something that happens to a person in the future. The examples are cataracts and leukemia are two types of somatic damage because those are happening to the person. Cataracts specifically happening to the lens of the eye, while leukemia is a malignant growth of or hyphen growth or proliferation of cells in the blood. On the other hand, Down syndrome is a type of genetic damage because it's due to damage to genetic cells, specifically a trisomy disorder on the 21st chromosome. So now we're on to Bouchon's chapter 30 on fundamental principles of radiobiology. In the terms of this chapter, we're going to start off by just a little review in the beginning by talking about what is ionizing radiation. It is defined as radiation that causes ionization, and that means removal of an electron from an atom. When we consider that ionization is going to result in damage, which leads to effects in the body, we're looking at ionization can occur with two types of radiation. Electromagnetic radiations are those that are waves, not particles, and they have energy, no matter. They travel at the speed of light, and they have electric and magnetic properties. And two of those that we use in the imaging sciences are X-rays and gamma rays that cause ionization. There are 7 types of electromagnetic radiations, but 2 of them are used in the imaging sciences. Particulate radiations can also cause ionization, and types that we specifically know of are alpha and beta particles. So alpha particles are known to be 20 times more damaging than beta particles. Alpha particles are something that are automatically emitted. There's alpha emission that occurs with radon. So when radon is inhaled into the lungs, it results in emission of alpha particles, and those alpha particles lead to lung cancer. So that's the effect that's measured, that can be measured in the person because they end up with lung cancer as a result of it. So when you're looking at alpha and beta particles in X-rays and X-ray and gamma rays, 3 of those have a radiation limiting factor of 1. X-ray, gamma rays, and beta particles. Alpha particles have a radiation limiting factor of 20. Therefore, they're 20 times more damaging and can lead to an effect, which is lung cancer. Now we're going to examine what's referred to as radiosensitivity that we talked about in the previous chapter. There's a law from 2 scientists that studied and theorized that radiosensitivity was a function of the metabolic state of the tissue being irradiated. And it's been referred to as this law, and I cannot pronounce these 2 French scientists' names, so I'm not even going to try to massacre it, but I call it the law of B and T. I'm going to guess it's Bergoni and Turbando. That's just a guess, but stab in the dark. So stem cells are radiosensitive, mature cells are radioresistant. So we talked about that in the previous chapter. Younger tissues and organs are radiosensitive, which kind of makes sense because that goes along with stem cells being young and mature cells being older. Then they stated that tissues with high metabolic activity are radiosensitive, meaning that they divide faster. It takes less time to get them from immature to mature. So they're going to be more radiosensitive. And a high proliferation rate for cells and a high growth rate results in an increased radiosensitivity. So anything with a high proliferation, or which is growth rate, is a high number of times you're going to have mitosis occurring or meiosis occurring is going to result in more sensitivity. So for example, lymphocytes have a higher proliferation rate than nerve cells do. if you're trying to grow nerve endings, or specifically nerve cells, it's going to take quite a while for that nerve ending to get feeling back if you have lost feeling to something, whereas lymphocytes divide very quickly. So this is an important law to note. Now, physical factors that affect radiosensitivity involve three things: LET, RBE, and what's referred to as protraction and fractionation. So we'll start with LET. LET is defined as the measure of the rate at which energy is transferred from ionizing radiation to soft tissue. You can think of this as absorption. LET is absorption. You're going to be transferring energy to soft tissue from that radiation. And it's a method of expressing radiation quality, which is energy, and determines the value of the WR. WR, going back to last quarter when we were reviewing, stands for radiation weighting factor. Radiation weighting factor. With high LET, there are more ionizations, which is resulting in more absorption. And this is just a numerical value here that you can calculate LET. The nice thing is we don't have to. The LET of diagnostic x-rays is a penguin in your textbook, which is why there's a little penguin down here, of diagnostic x-rays is approximately 3 kiloelectron volts per micrometer. So just to restate this again, LET can be thought of as absorption. RBE refers to the ability to produce biologic damage. So you can think of the RBE as the damage. RBE stands for relative biologic effectiveness, the ability to produce biologic damage. And so the definition given in your textbook also is just this equation. We nicely don't have to calculate it either, but the RBE for diagnostic x-rays is 1. Excuse me. So it's the dose of stated radiation necessary to produce a given effect divided by the dose of test radiation necessary to produce the same effect. So long story short, it's going to be in relation to LET. If you increase absorption, you're going to increase the effect. And so let's take a look at that. We're showing here with LET of different types of radiations, 0.2, then 0.3, and it goes on up to diagnostic x-rays being 3, and moving up to alpha particles, which are 100. LET referring to kiloelectron volt per micrometer. Here's that 3 that we talked about earlier. And then look at the RBE values. The RBEs are going up as well. So there's a directly proportional relationship between absorption and damage. So as absorption goes up, the damage goes up, and I think that probably makes sense there, hopefully. If you increase absorption, you're going to end up with more damage, since all damage begins with ionization to start with. So here's the relationship in a graph. As LET of radiation increases, the RBE, which is the ability to produce biologic damage, also increases. RBE is directly proportional to LET up to a point. And notice how it's going up. Here's Rollo here with cobalt, then X-rays, and going up, 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 up here to heavy nuclei, and then down. And it just goes to region of overkill. And what I like to think of that is like if you've already killed something, how can you kill it anymore? It's trying to overkill something. You can't kill something twice. It's dead. So here it is with the blanks filled in, just showing that as LET of radiation increases, the RBE is going to increase also. Increase, as I said before, ionization, we increase the damage. And we can also state in another way of referring to it is that RBE is directly proportional to LET up to a point, meaning as one goes up, the other goes up, and then they're inversely proportional. The good thing is we don't have to worry about the region of overkill because we don't use heavy nuclei. We're down here with X-rays for the most part. Alpha particles in some cases. We're going to examine the difference between gamma and X-rays, which have the same radiation weighting factor, the same LET, the same RBE, and the same OER, and compare them with alpha particles. Notice we left beta particles off the list just to simplify things. So to restate, gamma and X-rays are two types of electromagnetic radiations. compared to alpha particles, which are much, much more damaging and results in lung cancer. Let's take a look at what happens. So the LET for gamma and X-rays is low. The radiation weighting factor for them is 1, and the RBE is also low. So if there's low ionization, the number specifically for X-rays is 3. We don't have to worry about it for gamma. For an energy of the exact same type, it'd be low. It'd be 1 at 3 anyway. But we're going to get a value of 1 for the radiation weighting factor. There's no math you have to do at all for that. It's just a given from the NCRP. The WR stands for radiation weighting factor, and that's the weight of the radiation because it's basically due to its type of radiation. The RBE, which is the amount of damage we get, would then be low. On the other hand, if we say that alpha particles have a high LET, they have a radiation weighting factor given to us from the NCRP is 20, and they have a high rate of damage. And so high ionization, high absorption, high damage. We're going to talk about OBR shortly, it just happens to be in the same chart. The third and final factor that affects radiosensitivity is going to be protraction versus fractionation. If a dose is delivered over a long period of time rather than quickly, the effect of that dose is less. So stated differently, if the time of radiation is lengthened, a higher dose is required to produce the same effect. And this is just a basic statement from your textbook. So to shorten it up, protraction is a radiation dose delivered continuously, but at a lower dose rate. And so with this, it's called to be, or known to be called protracted. An example is if 6 gray was delivered at a rate of 3 minutes. So it'd be 2 gray per minute if you look at how long it took to do that. Or doing it at 6 gray, delivering it at a rate of 10 milligray per hour for 600 hours. So looking at which one would happen is that the first one, delivering it in 3 minutes, would be deadly for a mouse. But on the other hand, if you delivered it over a period of 600 hours, the mouse would survive. So it's a matter of lengthening the time that you deliver radiation, but shortening up that dose rate. And that's the second one here is referred to as protraction. But who wants to lie on a table for 600 hours if you are a patient and you're in radiation therapy? And how would you get a mouse to lie there for 600 hours either? So it's not likely to do that. What we have instead in radiation therapy is fractionation. And the word fractionation you can think of as in fractions, equal fractions. So radiation is delivered at the same dose in equal portions or fractions at regular intervals. We have 6 gray, the same amount, delivered in 2 gray per minute, but in 12 equal fractions of 500 milligray separated by 24 hours. What happens in radiation therapy or oncology? Well, we have recovery and repair that occurs between doses. If you look at their scheduling in radiation therapy, a person comes in at the exact same time every day for their treatment. They're there at 9 o'clock, Monday, Tuesday, Wednesday, Thursday, and Friday. They have 24 hours separated between each dose. They have the weekend even longer to give more time for recovery. And that is going to give equal treatment, the same effect that you have with protraction, but you're going to do it in intervals versus having someone lie there for 600 hours. Who could be there that long? And besides, they wouldn't feel too good either. So this gives more chance for higher doses to be used and for repair and recovery to occur between them. Another thing it affords is that we can use higher doses of radiation to treat cancer and get better remissions, and maybe more palliative care as well, depending upon what we're doing. Now, there are also biologic factors that affect radiosensitivity. The first factor is looking at oxygen. There are 2 parts to oxygen. I've got them written down as A and B. So A is on this slide, B will be on the next one. So biologic tissue is more sensitive in an aerobic state. What does that mean? Aerobic means what? It means with oxygen. So if you increase oxygen, you increase sensitivity, right? So it goes hand in hand. And that means that if you have medium sensitivity, the person's in a hypoxic state. Hypoxic is less oxygen or state with less oxygen or decreased. There's least sensitivity, radiosensitivity in an anoxic state, which means without oxygen. And then we can think of this as being using hyperbaric therapy and radiation therapy. I'm going to talk about two things. One's kind of morbid, but the second one is about radiation therapy, which makes it kind of interesting too. Is that if you think about, if you were trying to study radiation on cadavers, it wouldn't work. Why would that be? Because cadavers don't have oxygen in their tissues. So you couldn't just x-ray cadavers and expect the same result. There's more sensitivity with live tissue. I know it's kind of morbid, but it's just something to think about. So the second thing to look at is that, well, cancer itself. If you look at cancerous tissue, it's really cool to look at. If you could when you open a person, you look at the inside. I was able to see this one time, and I was just in amazement. But oxygen, cancer tissue next to good or normal tissue is there's a difference in color. Cancer has a grayish appearance, like a deadly appearance, where live tissue or the good tissues are pink and more like your normal coloring anyway. And so when you think about oxygen or the actual cancer itself, it doesn't have as much oxygen. It's kind of almost just without a big hypoxic, really. So we tend to use what's called hyperbaric oxygen in radiation oncology, where we increase the level of oxygen in the person's body to increase the effect. So just something that we can do. Kind of interesting thing, something to consider. Also, when you're looking at it, I've had several students over the years say, well, why they always say that when you open a person up during surgery, that the cancer really starts growing. And it really does have a low amount of oxygen. So you expose it to air, it's going to have a different growth rate. The second part, or B part to oxygen, is called OER. OER stands for Oxygen Enhancement Ratio. I didn't copy and paste that into this PowerPoint slide because I figured you would know what that stands for, but Oxygen Enhancement Ratio is the dose under anoxic conditions to produce a given effect divided by the dose under aerobic conditions to produce the same effect. Therefore, OER is LET dependent. What happens is that OER is high for low LET radiations. If you use X-rays or gamma rays or beta particles or something like that, where you have a low LET radiation, kind of like X-rays have an LET of 3. They're over here. They're going to have a high oxygen enhancement ratio. There's going to be a high enhancement for them. But if you use alpha particles, they don't care what they have a high LET, but they don't care what they hit. They're going to have the same effect. Alpha particles do not care what they hit. They're going to have a really high response regardless. So oxygen is going to enhance X-rays. It's going to enhance the effect. So OER is high for low LET radiations and decreases in value as LET increases. Therefore, you can state that they're inversely proportional. And you can say that with your graph also, that here's LET on the x-axis, OER on the y-axis, and they're inversely proportional as you continue increasing the LET. OER keeps going down. So alpha does not care what it hit. It's going to kill it anyway, kind of like a machine gun. This is the chart we looked at earlier with regard to radiosensitivity with LET, WR, and RBE, where we had completed the chart for gamma, X-rays, and alpha particles. Notice again that LET stands for absorption, RBE stands for damage. OER talks about the rate for oxygen enhancement. So thinking about gamma and X-rays versus alpha particles, which one doesn't care if oxygen is present? That will be alpha. So alpha particles have a low OER. They don't care if oxygen is there. Well, X-rays, the effect of X-rays in gamma will be increased or enhanced if there is oxygen present. So that's why they have a high OER. So there's an inversely proportional relationship between LET and OER. Age is the second one. This is a pretty interesting thing too. When you think about when you're first born, before birth, I mean, not even just born, but before birth, that's when you are the most sensitive to radiation. It's before birth. Then at birth, we're kind of close to old age. Notice that the chart kind of goes down and comes back up and cups up again, almost like a U, but not quite. It doesn't swing up as far at old age. So we're going to put these down as far as what from start to finish and use the exact wording from your textbook for the verbiage over here on the left-hand side. So taking a look at this, humans are most sensitive before birth, and that means when you're an embryo or a fetus. Then radiosensitivity decreases into adulthood, which means you're going to go from being an embryo to a fetus to being a newborn, then childhood, adolescence, and adulthood. And then in old age, it says aged on here, which is around 70, we become somewhat more sensitive. We're the most resistant, it shows at maturity, which is 50. It doesn't say the word maturity in here, it just has that in your textbook. So to write this out, ranking from highest to low radiosensitivity based on age, we would start off with being the most sensitive when you're an embryo, then a fetus, and then old age at age 70. So I put that old age in green there to remember that it swings up almost like a U, but not quite. Then it goes back to newborn, then childhood, then adolescence, and finally where they have the lowest radiosensitivity or the most resistance at age 50. Now, one thing you can think of when you look at this, I had a group of students last year do a project on it, and they wanted to know why old age, and that's and it comes down to one conclusion is that you just really can't reproduce and repair things as well as you could when you were younger. And at old age is when you become somewhat more sensitive. And I had made that conclusion in class, and they went ahead and proved it within looking at literature, within research that had been conducted. The third thing is looking at recovery. Recovery is known to be the addition of not just repair, but also repopulation. So if you think about it, if you get a cell that's damaged, it's going to repair itself, but then we have to also have repopulation occur to have cells that survived to undergo division to replace the cells that died. So that repopulation could be due to mitosis. We've got the repopulation going on from parent cells to daughter cells. Atrophy is when a tissue or organ shrinks and it receives a sufficient radiation dose that occurs because some cells die and disintegrate and are carried away as waste products. When you think about atrophy, if you've ever broken a bone, your arm is in a cast, or leg is in a cast, or whatever part's in a cast, there's going to be atrophy or shrinking of tissues or organs, depending upon to which we're referring. And that happens very commonly. So that atrophy also happens when you're talking about radiation damage. Number 4 refers to chemical agents. Chemical agents can modify the response of radiation on cells, tissues, and organs. And so to be effective, they must be present at the time of irradiation. Post-irradiation application does not usually occur and does not affect the degree of response. So radiosensitizers are referred to as agents that enhance the effect of radiation. Their examples could be halogenated pyrimidines, any type of vitamin K products is what we're looking at, and your author goes into some detail about that, in your textbook. And then radioprotective compounds are those that have usually a sulfohydryl group of sulfur and hydrogen bonds together, such as cysteine and cysteamine. There's hundreds of others that have been tested and found effective by a factor of 2, but the problem is that in mice they found that radioprotective agents tend to produce lethality, so they're found to be toxic. Therefore, sensitizers could be used to enhance the effect of radiation, but radioprotectors have not found human application because to be really effective, they're going to be toxic to us, and the protection would be worse than the radiation itself. The final thing to look at with regard to biologic factors is hormesis, and that's a factor in which we consider that a small amount of radiation. Hormesis is the final biological factor, and it's a separate, and there's a small body of radiobiologic evidence that suggests that a little bit of radiation is good for you. And it's stating that there are some studies, and there are out there, I've had students research this, that have shown that animals given low doses of radiation live longer than unirradiated controls. And so an explanation is that a little radiation stimulates hormonal and immune responses to other toxic environmental agents. Many non-radiation examples of hormesis can be found, and that is, for example, in high doses, fluorine is deadly, but in small quantities, it's great as a tooth preserver. Regardless of this, we do want to practice ALARA. Another non-radiation example is our vitamins. If you take a large amount of vitamins, it's going to make you sick, or could be deadly, depending upon the amount. But if you take a normal amount, then it's going to be good for you. Radiation dose-response relationships are the next and final topic for this chapter. So we're going to talk about how we've had various things occur as far as responses to radiation, in which we'd be able to classify items as either being what are called stochastic or deterministic. And so this research from looking at dose-response relationships has led us to know how we look at effects. So we're going to talk about these real specifically now. So we have mathematical and graphic functions relate to dose as a result of observed response. And so it's been used to look at designing therapeutic treatment routines for patients with cancer. It's looked at information that we've had from effects of low doses of radiation and even those from high doses. So it just depends upon what we've examined. A body of literature, a very large amount of literature is out there for atomic bomb survivors. And so that's a great deal of information. So it's significant for diagnostic radiology, DR standing for diagnostic radiology, because we want to know how dose and what the responses could be for us. So these relationships are going to be either linear or nonlinear. First of all, they're either going to be straight lines or curves. On the left-hand side of the page on the bottom, you can see where we have dose on the x-axis, just as we do on the right side, and the responses on the y-axis. In these examples here, whether we look at A, B, C, or D on the left-hand side, these are all linear relationships, meaning that they're a straight line. On the right-hand side, A, B, and C are curves. And so they're looking at whether there's no inflection point, or an inflection point as there is in C. It's just a matter of just differences between the relationship. The second thing we looked at is threshold versus non-threshold, and we're going to be defining these in the next slide. But non-threshold means that we're starting at a dose of 0. So A and B in both of these graphs are blue. A and B on both of them are both non-threshold. They began at 0. There's no dose. Even with A on the left-hand chart, where there's a straight line with an RN, that N stands for natural. It's a natural response. And the dose is still 0. Looking at the green and I guess another shade of green, yeah. Green and 2 shades of green, they both have D with small t's. And that is being threshold doses. So they have a certain dose that you're going to have to take to cause a response. We're going to be above 0. So let's take a look at those definitions next. The definitions I was referring to on the previous slide are those that are related to the dose-response relationships, and start off with the term linear. In the term linear, the response is directly proportional to the dose, and that's when the radiation dose is doubled, the response to radiation is also doubled. The second definition is that at which there's a non-threshold relationship. So any dose is expected to produce a response, and so that begins at 0. The third definition is a dose at which a response to an increasing X-ray intensity first occurs. The dose is above 0, and that definition is threshold. The fourth, and below that threshold, there's no response. And finally, the fourth one is a natural response, the response level, which in the previous diagram was labeled Rn. N as in natural, indicates that even without radiation exposure, the response occurs. So these are the 4 definitions to which I was just referring: linear, which is also called a straight, which you'd also think of as a straight line, versus nonlinear, not. Non-threshold starting at 0, threshold starting above 0, and natural response where there already is a response but there's no dose. And we'll take a look at those in terms of identifying them at this point. Before we get there, I wanted to state also that these lines are something that are your author uses the term extrapolated. So there are estimates taken when they're constructing a dose-response curve or dose-response relationship. And so they're looking at irradiating animals or looking at population groups that had doses of radiation to a certain area or to a particular part or something transpired, and then they're looking at the responses compared to what should have occurred in that population group. So I'll give you an example shortly. In these 4 curves, 4 lines I should say, A and B, the blue lines, are both linear, and so are C and D, of course, the green ones. The blue curves, on the other hand, the N stands for natural, so that's where there's a natural response, where B is there is no natural response. The dose starts at 0 as far as the dose and the response. Both A and B are what are called linear non-threshold. Non-threshold means the dose is 0. So A and B are both linear non-threshold. C and D are considered linear also because they're straight lines, but they're called threshold because they start up at a dose above 0. Looking at these 3 curves, A and B are nonlinear, and C is nonlinear as well. If looking at A and B, they are nonlinear non-threshold because they're not linear and they are at 0, so nonlinear non-threshold. C has 3 names. It's called a nonlinear threshold, hence the T in the dose, dt, and it's also called the sigmoid curve because it's shaped like the sigmoid colon, or a sigmoid, which is an S shape, and then also the letter S. S curve, sigmoid, and nonlinear threshold. In terms of biological effects and how they relate to the dose-response relationships, we're going to be taking a look at stochastic and deterministic effects. The first type is looking at stochastic. Stochastic effects follow a low radiation exposure and appear as a late radiation response. There is no threshold with stochastic effects. As the dose increases, the frequency of response in a population increases, which means it's going to be a linear relationship. Since there is no threshold and it is linear, we would call that following a linear non-threshold dose-response relationship. And examples that your author discusses as common ones are cancer, leukemia, or genetic effects. However, I just want to make sure that it's understood that skin cancer is an exception. So taking a look at the slide, it's that we have low radiation exposures, late responses. So an example would be in diagnostic radiology, we have low doses of radiation in comparison to radiation oncology. If we saw any type of effects, we would see them as late responses. So radiologists in the early 1900s up to the 1950s approximately had a late response of radiation referred to as lifespan shortening. They died younger than the rest of the population. They also developed leukemia, more commonly the rest of the population. They were low doses of radiation compared to an atomic bomb being set off, which didn't happen until the mid-40s, 1940s, or much lower responses of doses of radiation than other population groups had. However, they had a late response, meaning they just died younger than the rest of the population, or they developed leukemia, which takes 20 years approximately to manifest. As I said before, there is no threshold. Any dose could cause a response. There's no certain dose. As dose increases, the frequency increases, so it's a linear relationship. It's going to follow the picture. In the picture, we're looking at letter B, the light blue line, starting at 0 for both the dose and the response. So it's a linear non-threshold dose-response relationship. A would imply that there was a response without radiation. So you already are seeing cases. And the examples are leukemia, cancer except for skin cancer, and genetic effects. Deterministic effects follow a high radiation exposure and appear as an early radiation response. There is a threshold with a deterministic effect. As dose increases, the severity increases and then it drops off. So it's not linear, is what that statement is saying. It follows therefore a non-linear threshold dose-response relationship. And so therefore, we're looking at letter C as the example. There's as dose increases, severity of the response increases and then it drops off. And the relationship is non-linear, so it does not occur, plus there is a threshold dose. For example, it's going to take 2 gray of radiation all at one time to be received to get cataracts. You cannot get cataracts if you do not receive 2 gray of radiation. As far as radiographers are concerned, you will never see that type of dose of radiation, even over your lifetime, to even begin to develop cataracts. So working in radiology does not cause cataracts. A decrease in sperm count has occurred in volunteer convicts in a prison experiment that took place in Oregon many, many years ago. And in that case, there was a result of 100 milligray that resulted in a decrease in sperm count. So there are set numbers that if a person has at least a certain amount of radiation, they get a response, and we know what that number is. And if there is a number that's given, it is a threshold dose, and that means it's going to be a deterministic effect. There is a threshold. Characteristics of the stochastic and deterministic. This is in a chart in your textbook that's on page 487. So stochastic effects have a non-threshold dose-response relationship. They're linear, so they're called linear non-threshold. The increase of response increases with radiation dose, which means it's linear also. Deterministic is always going to be a threshold dose. It's always going to be nonlinear. So therefore, it's going to be nonlinear threshold. And the severity of response increases with increasing dose. So when you're looking at stochastic, it doesn't always happen. In American radiologists, they didn't always see leukemia, but there were more cases in that population groups compared to even British radiologists. And we'll talk about that in a couple chapters from now as to why. Also, they saw a greater incidence in American radiologists compared to other population groups just within like how many they would expect to see. Because there's a certain number of cases that are just expected in the regular population group. And then otherwise, deterministic, where it's the severity of response, that just means that if you have erythema and you have more dose, you're going to get more erythema. Kind of like you're out in the sun I'm out in the sun for 15 minutes, I'm going to start to, later in that evening, I'm going to see a response. But if I'm out in the sun for 2 hours, I'm going to be a lobster. So the severity of my response is going to increase my dose to the sun. And that's what you're looking at with deterministic. So now we're on to chapters 31 and 32, which is molecular and cellular effects of radiation. These 2 chapters were previously one chapter, and I've continued talking about them as one because unfortunately, doesn't really work well to talk about one without the other. So it makes a little bit more sense to do it this way. So so far we've been talking about cells and molecules, and we're eventually going to be getting to a human being, which is pretty cool. Talking about effects to people. Let's talk about X-rays hitting cells and what that effect might be. The first concept to talk about are direct and indirect effects. Direct effect, otherwise called direct action, is where an X-ray photon directly or just hits DNA and causes damage. So here's a double helix configuration for DNA. X-rays hit it, cause damage. Done. That's what a direct hit is. An indirect effect is where X-rays hit water or some other possible molecule. The reason why we talk about water as being an example is because it's the most prevalent in the human body. Then we produce ions and free radicals. Well, free radicals are going to be reactive. They're going to go hunt out DNA and cause a reaction. Therefore, we're looking at two different ways to get to the same effect. Taking a look at the true definitions is that with a direct effect, X-rays strike DNA and cause damage, otherwise called a hit. And with the second one, indirect effect, X-rays strike water, which produces ions and free radicals. The free radicals strike DNA and cause damage. The other name for the indirect effect is called radiolysis of water. So the other name for indirect effect is radiolysis of water, because X-rays are hitting water. Is it possible to hit something else? Yes, you could hit lipids, you could hit carbohydrates. It's just a lot more common to hit water because 80% of the body is comprised of water, like I said before. So radiolysis of water is another name for the indirect effect. It's a interaction of radiation with water and results in the formation of free radicals and ions. So taking a look at this picture, we're looking at X-rays struck water. It breaks apart into ions, which are positively charged or negatively charged. And then they eventually break down into ions and free radicals. So the formation is of ions and free radicals. Ions are denoted with a plus or minus sign. There's H plus or OH minus. This is a hydrogen ion. This is a hydroxyl ion. Or free radicals, which have an asterisk mark. An asterisk denotes it's a free radical. A free radical is going to be very damaging, and we'll take a look at that definition here in just a moment. But with that, it's going to have OH asterisk, which is hydroxyl radical, free radical, or H asterisk, which is a hydrogen free radical. The definition of a free radical is on page 495 of Bouchon's textbook. It's an uncharged molecule that contains a single unpaired electron in its outer shell. Free radicals are highly reactive. They are unstable and therefore exist with a lifetime of less than 1 millisecond. During that time, however, they are capable of diffusion to the cell and interaction at a distant site. Free radicals contain excess energy that can be transferred to other molecules to disrupt bonds and produce point lesions at some distance from the initial ionizing event. So it also explains how the H asterisk and the OH asterisk are not the only free radicals that are produced in the radiolysis of water. There are others too. And he goes on to explain how you can form what are called hydroperoxyl free radicals and even organic free radical formations too. So there's other things that can be produced. But the free radicals are what are so damaging. There are three reactions that it can occur that happen with the indirect effect. We have ions combine when the X-rays hit water. They can combine to form a stable water molecule. No damage occurs if you have water forming. If you have free radicals, they can migrate to DNA and cause a hit. That produces damage, and that's the idea of the indirect effect. The third thing that can happen is what is called free radicals combine with other molecules to form a toxic substance or agent. And that's where we get what's called hydrogen peroxide. Hydrogen peroxide can migrate to a DNA molecule and cause a hit, and it produces biological damage. So the free radical is going to combine, two free radicals come together, and they combine to form H2O2, and that is hydrogen peroxide. And if you produce hydrogen peroxide in your body, that is toxic. So two of the three things we just talked about cause damage. The second one and the third one cause damage. Free radicals cause damage, and toxic substances or hydrogen peroxide cause damage. Ions do not. No damage occurs. From the direct and the indirect effects, continuing on with that discussion, the principal effect on humans is indirect. I'm going to talk about why. First of all, when you consider that 80% of the body is comprised of water, you're going to have a lot more indirect actions or effects occurring. So the human body is comprised of 80% water and less than 1% is comprised of DNA. So since the indirect effect occurs more often, that's going to be your principal effect. Also, the indirect effects are amplified because you're not just going to have one free radical formed, you're going to have two out of each interaction. And plus you can have more that can transpire from there, or toxic substances can occur rather than just having one X-ray hitting DNA. The next thing we have to look at is irradiation of macromolecules. In terms of irradiation, we have two types. We can have in vitro, which is outside the body, and in vivo is inside the body. In vivo means live, so it means inside the body. Well, where do you have oxygen in terms of inside of your body or outside of your body? It's inside of your body. So macromolecules are more radiosensitive when you have them and you x-ray them when they're in somebody than whether they're sitting on a table and then somebody has maybe a biopsy that was taken and it's the tissue specimen sitting on a table. There's going to be more radiation damage inside the person or more effects in the person than that tissue sample lying on a table with no oxygen. Now we have irradiation of DNA, whether it happens with the direct or indirect, it doesn't matter, whichever effect it occurs with. There's three responses going to happen. So it doesn't matter how it happened, it's just when the DNA is hit. We can have what's called main chain scission, which is just like taking scissors and cutting a side rail of DNA. Main chain scission again, scissors, with cutting both side rails is the second one. The third one is what's called main chain scission and subsequent cross-linking. Cross-linking is when you join one molecule joins with another one. Well, why would that happen? Why would we have cross-linking occur? Well, think about what DNA is comprised of. DNA has deoxyribose sugar. What else is in your body? Water. What happens when you put sugar and water together? You drink a Coke or you drink a Pepsi, it's got what? It's sticky. So what happens is cross-linking is when side structures become sticky from when they've been cut, and therefore the two start to come together. So it's like if your hand gets sticky with crazy glue and you touch your fingers together, your fingers stick together. So that's what happens with cross-linking. You can also get rung breakage that occurs separation of bases. So if you think about DNA being like a ladder, the side rails are where you got the scission happening, or the scissoring. If you break a rung of a ladder, which is where you step, you can separate your bases, which are on the rung of the ladder. And finally, you could change or lose a base, which would be highly detrimental. So let's take a look at a picture. So there are 5 responses from left to right. A is where we have main chain scission or cutting a side rail of DNA, one side rail. B is looking at cutting both side rails, which would potentially, if you cut at the exact same level, if it wasn't here and clear up here, but if you cut at the exact same level, we would break the DNA macromolecule into 2 pieces. The third is showing where we have a DNA macromolecule, but there was main chain scission, which caused stickiness, kind of like that superglue example I gave, or pop being Coke or Pepsi being sticky, and then you touch it with something else. So you touch your 2 fingers together. Not that I've never done that before. And you've got another DNA macromolecule kind of stuck to one of them. So two are stuck together. So that's main chain scission with subsequent cross-linking. D, which is the fourth example, is going to be just a rung breakage. They didn't give the best picture on this, unfortunately, but you can see where they've just lost some rungs. But yeah, it's just rung breakage. So if they were just to show a little break in a rung would be better. And the last one is where you've got a certain setup of your base, certain set for how your bases are supposed to be on a rung, on each rung of your ladders. Well, what if you lose a base? You completely lost it, or you've got the wrong, they've gotten mixed up. So normally, adenine and thymine sit on the same rung, and cytosine and guanine sit in the same rung. So if you rearrange your genes, or you were to lose one of those bases, you'd have a major issue. So that one last part is this one over here, number 5. This fifth response may cause irreparable damage. The first 4 can be repaired. You might be able to repair main chain scission of 2 cytosines, even main chain scission with cross-linking, or even a rung breakage, but it's very difficult, may not be possible to repair the loss or change of a base, which is the 5th one. The most sensitive or radiosensitive molecule in the human body is DNA. DNA is also the most important molecule in the human body. So DNA is going to make up your chromosomes, which makes up your cells, which makes up you as a person. Everybody is a different human being because they have different DNA. So we share DNA, there's crossover from our parents, so genetic traits do cross down, but we're not a replica of our parents in most cases. So, excuse me. So you may have traits that are similar, but not an identical being. There's possibilities of visible DNA damage, highly possible damages that are possible. We have to damage a lot of DNA to cause this to occur. I mean, a chromosome contains miles of DNA. So to see a visible change under a microscope takes quite a bit. Right? So if a visible aberration occurs, means that there's a lot of damage. The first chromosome that you see in this picture, in A, is showing after the S phase. We already had duplication or replication of DNA, and so this is normal. In B, we have what's called terminal deletion, where we lost some DNA here at this area and down here at this area, so we call that terminal deletion, otherwise called chromatid deletion, because this is a chromatid, and this is a chromatid. This formation happens because of main chain scission and that stickiness, that cross-linking, we get what's called dicentric formation. And the last one is also from the cross-linking where the DNA ends up going into a circle, which gives it a ring formation. These last two you can call dicentric chromosome, and the last one you can call ring dichromosome also. So they can be dicentric formation or chromosome, and ring formation or ring chromosome. But these are visible changes that you can see under a microscope of a person's DNA. Specifically chromosomes, not DNA, within their chromosomes. Irradiation of DNA is going to result in 3 observable effects in humans. This is within a person now. We're not looking under a microscope anymore. It can cause malignant disease, which means cancer. The second thing is cell death. And the third thing would be genetic effects. So let's talk about those a little bit further. Malignant disease, like I said, was cancer. So the cause of stochastic effects, which could be cancer, is damage to DNA results in abnormal metabolic activity. That means there's uncontrolled rapid proliferation of cells is the principal characteristic of radiation-induced malignant disease. So Marie Curie, who developed radiation-induced leukemia, and her daughter Irene had the exact same effect from working with radioactive materials. They both had abnormal proliferation or growth of cells, specifically of white blood cells. They both, in that characteristic, had that high metabolic activity of those cells. That resulted in a stochastic effect. Number 2, what is the cause of deterministic effects? So deterministic effect is an example of one in which there's such as decrease in sperm count. There's cataract formation. So there's some type of damage to DNA that leads to cell death. If enough of the same type of cell responds, then a tissue or organ can be destroyed. So cataracts result in opacity of the lens of the eye, so that tissue is being destroyed. In the case of decrease in sperm count, there's a loss in sperm count within the male. It can also be that you could have a loss of menstruation within a female with the exact same radiation dose. That was studied as well. So that's the cause of deterministic effects, is cell death. The third type of effect is the cause of genetic effects, and that's damage to DNA that occurs within a germ cell. So the only way you can get a genetic effect is if you have damage to radiation in a germ cell, or otherwise called a reproductive cell. So this has to have occurred in a spermatogonium, or somewhere along the line of whether it's spermatogonium, spermatocyte, spermatid, or spermatozoa, whatever stage of the cycle it takes, and the effect is going to be seen in future generations, or maybe even a later one. We are still watching for Down syndrome to occur in the A-bomb survivor population group. A-bomb survivors are anticipated, or didn't have it themselves because they were the ones that survived the radiation exposure, but we'll talk in the 2 chapters ahead as to why that's difficult to see. We have observed it in mice and fruit flies, though. So it occurs in a germ cell. The target theory is the concept of a key sensitive molecule, or sensitive key molecule, and that molecule is DNA. In order for a cell to die following radiation exposure, its target molecule must be inactivated. So radiation does not look for DNA, it's going to interact in a random fashion. So the theory is that in order to kill a cell, you have to kill or hit the DNA. Okay, we're on to chapter 33 on deterministic effects. This is where we start talking about people and examples on people, so I think it's a little bit more interesting. It's kind of sad in one way because we have had a number of cases of different effects that have occurred, and anytime you'd like to hear more about those, I can share them. I was just trying to keep this lesson short as possible. What you're looking at here is a person's head in which they have a burn. It's not necessarily a radiation burn, but I just thought I'd show you, just to be kind of interesting. It could have been erythema. Early somatic effects are radiation responses in humans that occur within a few days to months. Keep in mind that early effects occur within a short period of time, such as days and months, and that somatic effects occur to the person who was exposed, or to the body part that was exposed. These effects are pretty rare in diagnostic radiology because we rarely have high doses of radiation in our area. Deterministic effects are what they're usually called, but these are effects that exhibit increasing severity with increasing radiation dose. An example would be erythema, which has a threshold dose of 2 gray. It takes at least 2 gray to cause erythema. The effect is going to be higher, or going to have more redness, if there is a higher dose, such as 3 gray. And I'll give an example shortly. The dose-response relationship, therefore, is usually nonlinear threshold for deterministic effects. In terms of threshold doses, there are threshold doses, which means the lowest dose that's going to take, but it will cause a response. And we would hope that we wouldn't cause these things with any dose of radiation. So when you're looking at death, a whole body exposure can cause death at a dose of 2 gray. Hematologic depression, which is a decrease in white blood cells, specifically lymphocytes, can occur at 250 milligray with the whole body being exposed. Now, if you just look at a small field, just column it down, skin erythema can occur at 2 gray, Epilation, which is loss of hair, can occur with 3 gray, and gonadal dysfunction, irradiated just to the gonads, can occur at 100 milligray. In males, it'd be a loss of sperm count, and with females, it'd be a loss of decrease in menstruation or loss of menstruation. As far as the stages of ARS are concerned, we kind of stated them on the previous slide, but I wanted to provide a little bit more information. I took a couple pages from the chapter and put material into a form of a chart that might be helpful for you. So to start with the prodromal period, that's a period that's going to be at doses that are above approximately 1 gray that's delivered to the whole body. There are going to be signs of symptoms of radiation sickness that can appear within minutes to hours. And these symptoms of early radiation sickness often take the form of nausea, vomiting, diarrhea, and a reduction in the white blood cells, the peripheral blood, otherwise referred to as leukopenia. This is the immediate response of radiation sickness. And this period of time is going to last from a few hours to a couple of days. The severity of symptoms is dose-related, so at doses in excess of 10 gray, symptoms can be violent. At still higher doses, the duration of the prodromal period becomes shorter, as it's difficult to separate the prodromal period from the period of what's called manifest illness 2 stages later. Now, the latent period is the next stage that a person undergoes. After the period of initial radiation sickness, called the prodromal period, a period of apparent well-being occurs. This is called the latent period. This latent period extends from hours or less at doses in excess of 10 gray to sometimes weeks at doses that are lower, of 1 to 5 gray. The latent period is the time after the exposure during which there is no sign of radiation sickness, and the person generally feels well. In fact, they indicate there's an early recovery, but it can be misleading because there's no indication of where the radiation illness and sickness is going to go. The manifest illness has been noted by many of healthcare providers as being the period during which the symptoms of the true syndrome occur. And there's 3 separate syndromes that can occur. There's either hematologic, otherwise called hemoptytic, which is dealing with just the blood. There's the GI syndrome, dealing with the gastrointestinal system, and CNS syndrome, which is dealing with just the central nervous system. But these are the effects of the exposure that are truly seen. As far as the last stage is that there's recovery or death. The primary effect of the acute radiation syndrome is shortening of the lifespan. As far as recovery or death, there is a possibility for a person to recover from the hematologic syndrome. So that is a possibility, or the person could die if they just have a weakened immune system or their dose is just a little bit higher than like around 9 or 10 gray. If the person undergoes or goes into the GI or the central CNS syndromes, then there's only the possibility will be death. Let's take a look at this in more detail. I think it'll help when we get to the next slide. So the next slide, what I did is I took another couple of pages and I put the information into a chart for you. Well, I actually did this many years ago, so I keep redoing it, but it just gives the information about the hematologic syndrome, exactly what dose, which it occurs, how much dose it takes for that syndrome to occur. So after the dose is estimated that the person had, they can estimate which of the syndromes are going to go through. And then they go through the prodromal first, then the latent, then the manifest illness, and then finally the death or the recovery. As mentioned before, recovery is only possible if the person receives a dose so that they only go the hematologic syndrome. If the dose is high enough to where they go undergo the GI syndrome or CNS, then they will die definitely die. And then we'll talk about the mean survival time. So to start off with, the dose to go into the hematologic syndrome is 2 to 10 gray. So if a person received 5 gray, they would definitely undergo the hematologic syndrome. Doses have always been able to be estimated as far as these are usually accidents that have occurred or the person was poisoned once with a Russian spy was poisoned. So that different things have happened where we've seen people go through radiation sickness and as early as just a decade ago, as recent as a decade ago. The prodromal period is the first stage the person undergoes with the hematologic syndrome. They're going to have mild symptoms of nausea, vomiting, and diarrhea. They're going to be tired or fatigued, and they're going to have leukopenia. As described before, that means a decrease in white blood count. This prodromal period is going to occur in a matter of a few hours. They will be fine for a couple hours, and then they're going to head into this period where they're going to be sick, and it can last for several days. After that same person in the hematologic syndrome has that dose and they go into the prodromal period, they're then going to go into what's called a latent period, and that's 4 weeks in length. Their cells in their peripheral blood will continue to decline. That can be watched by doctors or observed under, using lab work, but they're going to feel overall pretty well. Then the manifest illness will hit. They're going to have a decrease in their white blood cells, red blood cells, and platelets, so all blood cells are affected. They're going to have vomiting and mild diarrhea again. Malaise, which is just being very tired and lethargic, and they're going to have a fever. And these are all symptomatic of hematologic syndrome of the blood. Recovery is possible with this. If the dose is not lethal, they're going to have recovery in 2 to 4 weeks, but it may take as long as 6 months for full recovery. So people who have had this amount of dose have recovered. Some have died, and when they did, they died because of generalized infection, electrolyte imbalance, and dehydration. The total length of time, if you're going to die, that you would survive or live would be 10 to 60 days with this particular syndrome. So they could live from a week and a half to 2 months and then still die, or they may have full recovery after 2 months. These are actual textbook scenarios, research scenarios that have occurred. So people have been in these different types of syndromes due to radiation exposures, and we've observed them. So that's how we know exactly what's going to happen. The GI syndrome is going to occur with a dose received to the whole body of 10 to 50 gray. So keep in mind, these are whole body doses. These are accidents that have happened in the laboratory. It happened in New Mexico, for example. For one of them I can think about that it was Fat Man, Little Boy was a movie that was based on, and showed even him using the inverse square law to find out who was going to survive and who would die in the room in which there was an accident that had happened. So he was the only person that died, and he calculated he was going to die, but it took him, he was able to go and make, kind of settle his affairs before he was actually sick. Didn't have much time, but he had a few hours. So the dose for GI syndrome is 10 to 50 gray, and you have vomiting and diarrhea, and within a few hours of the exposure. So in this case, the actor was able to show that he was able to go and talk with his girlfriend, and kind of settle some things for himself before he got sick. And then he had 3 to 5 days where he felt good for latent periods, so they showed that in the movie as well. Not so much in the movie, but they kind of did displayed a little bit. And then manifest illness is where there was the damage to the hematologic syndrome system will occur, but it's much more severe. So they're going to have a decrease in all their blood cells. They're going to have more vomiting and diarrhea. They're going to have anorexia, which, because they're not going to feel like eating, of course, become lethargic. And then they're going to have diarrhea leading to loose and watery bloody stools. And that's where the problem comes in. Their death or recovery is that no recovery is possible. And that's where the actor in this case, his name was John Cusack, had calculated for the character that he was playing. It was based on a true story. That he was, in real life, his name was Louis Staten, something like that. Anyway, the actor showing that he was not going to be the only person not surviving, that the others were at a greater distance. So they underwent the hematologic syndrome. The death occurred primarily because of severe damage to the intestinal crypt cells. If they have watery and bloody stools, eventually the lining of their intestinal crypt cells are destroyed. And that's the reason why they're going to die. They can't absorb the moisture or nutrients from their own food. And they die within 4 to 10 days from the original exposure, the initial exposure. The CNS syndrome is where a person's been exposed to greater than 50 gray, and that has occurred as well. And they kept a man alive longer than what they should have, and that was the one within the last decade. And he begged them to let him go, but they tried everything they could to keep the man alive. There's severe nausea and vomiting within a few minutes of the exposure. They're extremely nervous and confused, and they have a complaint of a burning sensation of the skin. In his case, his skin was completely off, and it was down to like a bloody level, to the basal cell layer, below the basal cell layer, excuse me. So they had him up, like tied up, so his skin wasn't touching the surface of the bed or any type of sheet or anything. They had a burning sensation of the skin. They lose vision and consciousness within the first hour. But then for 12 hours, they come back, and they sometimes may subside or disappear for that period of time. And then the symptoms come back suddenly with extreme severity. They become disoriented, lose muscle coordination, have dyspnea, go into convulsive seizures, have loss of equilibrium, ataxia, lethargy, and lapse into a coma, and die within 3 days. Well, this is what with this particular person, to which I'm referring, they did everything possible with medical intervention to keep him alive longer than the 3 days. And he was in horrendous pain, and they couldn't use enough morphine to keep him calm. So they tortured the man, essentially. As far as death and recovery, the outcome has always been a death within 3 days, except that one exception where they had extreme medical intervention. No recovery is possible, and the cause of death was eventually elevated fluid content in the brain. But we've had more than one person die from this too. There's been a lot of accidents that have happened. Okay, so LD56 is referring to lethal dose, LD, and the 50/60 is looking at a percentage and then a number of days. So in a previous edition, several editions back of textbooks, this was not LD50/60, it had different numbers. It was LD50/30. So if you see an old question from a mock registry, not necessarily one I give because I try to fix everything that's old on those, but if you see one in Correct Tech or maybe in Red Tech Boot Camp or something, you may possibly see one where there's some different numbers. But the correct numbers that have been using for the last 10 years has been LD50/60. So LD50/60 is a measure of acute lethality, which is death, and it's a whole body radiation dose that causes 50% of the people to die within 60 days. I left that blank there so you can make sure that you know the difference between what the percentage and the days are. So it causes 50% of the people. The first number is the percentage, and the second number is the number of days. Now, the LD50/60 they found in humans is different than in other mammals. They've tested this pretty thoroughly, even down to comparing us to pigs, dogs, newts, cockroaches. I mean, it's unbelievable, this number of animals or mammals that they've tried. But ours is 3.5 gray is the amount of radiation it would take. So if 20 people were in a room and we received 2.5 gray radiation to our whole bodies all at one time due to some type of explosion, and there was gamma radiation and it was dispersed, then half the people in that room would die in 60 days. You can see that it's a non-linear dose-response relationship, so non-linear, and it would be a threshold because that's the threshold dose it would take to cause the death. Local tissue damage is when only a part of the body or portion of the body is irradiated. So it's not the whole body like we've been talking about so far. It's just a small collimated dose. So in this case, it takes a higher dose to produce a response. So for example, We could look at gonadal issues, such as a loss of menstruation. It's going to take collimation down to just the gonads and only radiate those to look at radiation doses. It's going to take a pretty high dose to cause it. So we're not going to suffer from a decrease in menstruation or decrease in sperm count or anything like that as far as being radiation workers. We wouldn't possibly even get that much of a dose, that high of a dose, and nor do we cause that in our patients. I'll explain where this has occurred. So the effect is cell death, which results in atrophy or recovery, and the local tissue response depends upon the radiosensitivity of the material in which you're radiating. So how intrinsically how radiosensitive is that particular cell or organ. And the kinetics is cell proliferation and maturation, meaning that how often do the cells regrow and mature? How fast does it happen? When looking at local tissue that can be affected immediately, we're talking about the skin in terms of maybe things like erythema, the gonads with changes in menstruation or sperm count, and also bone marrow in terms of affecting the blood counts. The dose-response relationship has always been a nonlinear threshold where there is a certain dose it takes to cause a response. There is a minimum dose to cause a deterministic response, as I just said. So when the the threshold dose is exceeded, the severity response is going to increase with increasing dose. So if it's going to be redness of the skin, it's going to be a darker redness or deeper redness if with higher dose. And it's going to be random. There's no absolute natural thing because it's not a linear response. It's nonlinear. The most sensitive cells of the skin are in the basal cell layer. It's the layer right beneath the epidermis, of course. And as far as effects in the skin, we have seen these happen with orthovoltage X-rays. Those are orthovoltage X-rays are those of ranging from 200 to 300 kbp. Previous use, they were used in therapy of deep-lying tissues. The unfortunate thing is it was used in radiation therapy where the skin received a higher dose than the higher radiation doses than the tumor did. The skin responses to those patients were erythema and desquamation. Desquamation is ulceration and shedding of the skin, kind of like shedding like a snake. Superficial X-rays were previously used in therapy of superficial tissues, which makes sense why they're called superficial X-rays. There are different sources. One says that it's the X-rays were vary between 50 to 100 kbp. The other one said between 50 to 150. So I just put the total range for the kbp amount. But the skin response was just erythema. Not that that was bad, but it was just different levels of erythema. Another type of X-rays used were Grenz rays, and those were rays that were between 10 to 20 kbp. Their specific use was in dermatology and typically with children to treat ringworm. The skin response was epilation or loss of hair within these children. As far as potential responses of skin from high-dose fluoroscopy, this was looking perhaps at a cardiac cath lab, angiography, different things like that. So early transient erythema is going to occur as far as the earliest stage of redness of the skin would be 2 gray. Now that's a pretty high dose, 2 gray. Approximate onset is going to be hours. A person could potentially leave a radiology room, fluoroscopic procedure, and hours from now, or at that point, would then develop the erythema. Kind of like you go out in the sun and you don't have a sunburn instantly, it happens later on. Main erythema would be where you have a severe redness, and that would take at least 6 gray for it to occur, and the onset wouldn't be for 10 days. Temporary epilation would be temporary hair loss, and that would take a minimum of 3 gray, and it takes 3 weeks to see that occur. That happened with a child who was irradiated 150 times in CTs of the head, and he had temporary epilation. But it was at great risk due to the number of exposures he received for having at very high risk for in the future for having cataracts. Permanent epilation would be 7 gray. so permanent hair loss at 3 weeks also. And the moist desquamation would be moist ulceration with shedding down the road with, it would take 15 gray, and the onset would be 4 weeks. Have we seen any erythema in thoracoscopy? Yes, there have been 2 cases in California that have been studied and have been documented. And the results were directly found to be related to the procedures that were performed. The problem is that some people leave and they don't think to go back about it, or they don't realize it was related to the procedure. So in these cases, the patients were found to be, it was directly related to the room, thoracoscopic room. As far as the gonads are concerned, most of the information has been gained from animal experiments. It was also gained from radiotherapy patients, meaning that a patient had to have radiation directly to the gonads for a particular type of cancer, or a radiation accident victim that was accidentally irradiated. A common population group that was irradiated were volunteer convicts in a prison in Washington State, or was it Oregon? Washington or Oregon, sorry. Anyway, they supposedly volunteered to be willing participants in an experiment, and they sustained high levels of radiation exposure, and eventually many of them became permanently sterilized. What they found from these groups of people is in both males and females, we get the same result. 100 milligray will delay or suppress menstruation, and 2 gray will result in temporary infertility. 5 gray will result in permanent sterility. Hematologic effects, minimum whole body dose to produce a measurable hematologic depression is 250 milligray. That's a whole body exposure. The first cells affected are lymphocytes. And lymphopenia, which is a decrease in white blood cells, and specifically lymphocytes, will occur within minutes or hours following the exposure, but they're slow to recover. Cytogenetic effects are the study of the genetics of cells, specifically cell chromosomes. We can look with early effects under a microscope at a person's at their chromosomes, and we can see the visible derangements of the chromosomes themselves. If you look in this picture, we can see where they've lost part of an end of their chromosome. In this case here, they lost the end of this chromosome, and here's a little chromosome that's stuck out there. These are called isochromatids, where there's just little dots that are left out in the cytoplasm. Here's a dicentric formation we talked about, and then the ring formation, here's one down here, and here's one over here. So there's different formations that can occur, transpire. Here's a very good one for the terminal deletion. So the end of the chromatid is off, and then they're located over here in the cytoplasm. There are 2 types of chromosome aberrations. There's single hit and multi-hit. Literally, the chromosome was hit once or more than once. Single hit chromosome aberrations follow that linear non-threshold dose-response relationship, and they start off at 0, so it's non-threshold in both cases. The multi-hit follows a non-linear dose-response relationship, so non-linear, non-threshold. So we'll write that down on here in just a moment. So single hits, where we have single hits of a chromosome, they get hit once, the end of the chromosome pops off, and we're left with little isochromatids. Those are fragments of a chromosome that do not have a centromere. And so the response is linear, non-threshold. The other possibility is more than one hit in the same chromosome, and the result can be a ring formation or a dicentric formation. So either one of these, ring chromosome, dicentric chromosome, or we can have what's called reciprocal translocations. That's down here in the bottom. Notice that there's a pink chromosome and there's a green chromosome. They're hit prior to, like in the G1 phase, prior to S. And then after the S phase, each one of these has, at that point, had the top come off or the end come off, the isochromatid, and they switch off and they adhere to the opposite chromosome. So they rearrange the genes but do not lose genetic information. So that can cause some issues for genetics. It's nonlinear, non-threshold as far as any of these results. that are multi-hit. Okay, in the last chapter is chapter 34 on stochastic effects. We'll begin. In this chapter, we're looking at stochastic again, and then we'll review deterministic. Just a little review is that stochastic effects are resulting from an initial radiation that happened and the cells retain a memory, is another way to think about it. We're going to have a chance or probability for response that increases as a result of increasing dose, so it's directly proportional with a linear response. Any dose can result in a response, so it's non-threshold. It's due to low doses happening over a long period, delivered intermittently over a long period of time. So American radiologists were a common person studied for these stochastic effects. Just to review, deterministic effects had a threshold. They had that severity of the damage increase like erythema, and we had high radiation doses for them. With effects of radiation, we're looking at radiation protection guidelines are based on stochastic effects for diagnostic radiology. Therefore, they're based on the linear non-threshold dose-response relationship where any dose can cause a response. We want to be safe and make sure everyone is safe. We've had epidemiologic studies of people exposed to radiation, and it's difficult to really examine whether the responses were due to the radiation because the dose was generally not known, but usually presumed to be low, and frequency of responses from diagnostic radiology are extremely low. So let's talk about cataracts. Cataracts are not a typical type thing. They're unusual. They actually follow deterministic. Some background information. We had a scientist by the name of E.O. Lawrence who developed the first cyclotron back in 1932. The purpose was, or intention was, to study nuclear structure. The cyclotron is used today to produce radiopharmaceuticals for nuclear medicine and for PET. So a very different purpose today. After Lawrence developed the cyclotron in 1932, the first case of cataracts seen in cyclotron physicists was in 1949. At that time, the cyclotron physicists were in one room, and there was a high beam of particles extracted through a tube and steered by electromagnets onto a material in an adjacent room. And unfortunately, they looked directly into the beam when they did so. As a result, they did have excessive exposure to the lens of the eye and resulted in having cataracts. And since there weren't that many cyclotron physicists at that time, they found that by 1960 they had several hundred cases that were reported in this small population group. They found that the radiation-induced cataracts occurred on the posterior pole of the lens of the eye. They had 4 conclusions that they drew from this, is that first of all, that the radiosensitivity of the eye was age-dependent, meaning that as the physicist was older, as the age of the person was increased, then the radiation effect did increase also, and the latent period became shorter. So they were looking at the idea that in old age, we become more sensitive. They found that the latent period was actually, they didn't have any symptoms for 5 to 30 years in some people, but the average was 15 years. And they did use high LET radiation, such as neutrons and protons, so it was necessary to have that high RBE to produce cataracts. We don't have that in diagnostic radiology. They also found that there were some pretty high doses that it took to cause cataracts, and by no means do we even come close to that in diagnostic radiology. 2 grays required to adjust the cataract, to adjust the lens of the eye itself, I should say, to cause cataracts following a one-time exposure or acute exposure. For a fractionated exposure, it would take 10 gray. That's over fractions of time or long periods of time. So for us to get cataracts, it would be fractionated, and we will never reach 10 gray in our lifetimes. So the dose-response relationship is linear threshold, because there is a threshold dose. Our occupational exposures to the lens of the eye are too low to require protective lens shielding, but we can still wear them. It's not that it's not impossible, and we'll talk about that in radiation protection. And it's nearly impossible for us to reach the threshold dose because we're not going to receive that high of a dose since we have dose limits that are imposed on us. Some considerations in CT for patients is that lens dose can be 50 milligray. So could a person eventually get to that fractionated exposure? That would be possible if they received an excessive number of CTs of the head. Protective lens shields are not normally required for CTs, scans of the head, but it is common to modify the exam, such as angling the gantry, to reduce the dose to the lens of the eye. Lifespan shortening was examined in radiologists specifically and found in no other population groups. The exact cause of death was just dying early. The dose-response relationship within the group was found to be linear non-threshold, and scientists examined or determined that from their research that at worst, humans can expect a reduced lifespan of approximately 10 days for every 10 milligray in tissue received. There's a table showing you just what are some risky conditions or specific things that have occurred, even speed limit changes or airplane crashes or cancer, working as a coal miner, being unmarried or being male versus female can cause as far as resulting in days of life lost. Now, the incidence of being a radiation worker of 12 days lost of life have not transpired in quite a few decades. So we'll take a look at the statistics in the next slide. In the 1930s, radiologists were found to die approximately 5 years younger than the general population. We can also consider that radiologists receive higher doses of radiation compared to the radiation worker or the radiographer specifically. That difference shrunk to zero by 1965 and has never resurfaced again. But it was definitely an issue for 35 years. Radiation-induced malignancy is our next to the last one of our last topics. We're going to be talking about how there's a natural incidence of death from malignant disease. It's approximately 20%, so it's not just due to radiation, of course. And cancer is the most important somatic effect caused by exposure to ionizing radiation. It's been the most studied one too. Leukemia, since it's such a rare disease, made analysis of that type of malignancy easier. There's a lot more data on leukemia than the other types of cancers. So types of cancer studied, as you can see here, there's a pretty good list there. We'll start off with leukemia. So it ends up being kind of a history lesson as far as what has happened with each one of these, and I'm going to keep the history kind of short because the registry is not going to test you as much on history. But leukemia does show a dose-response relationship of linear non-threshold. It has a latent period of 4 to 7 years is what they found, and that the at-risk period of watching blood counts increase, and then finally the person going into full-blown leukemia has been 20 years if it's radiation-induced. Marie Curie was a person who had radiation-induced leukemia, so did her daughter Irene. Other population groups included A-bomb survivors was the greatest one studied. Early American radiologists was a huge group. There were a lot more physicists who were radiologists and vice versa in Great Britain. So in Great Britain, the early radiologists weren't as exposed to radiation as our American radiologists were. Ankylosing spondylitis patients who received a large dose of radiation and they were able to stand upright, but they ended up with leukemia 20 years down the road. Radium watchdog painters, children who received superficial X-ray treatments, some other radiotherapy patients, meaning they had radiation treatments and then they developed leukemia 20 years down the road, and then children irradiated while in utero, especially in the A-bomb survivor group. So on the left-hand side is a picture from state-of-the-art equipment from 1933 at the Mayo Clinic. And you can see where the radiologist is sitting directly in front of the patient looking into a screen, and there's no image intensifier. And here's the controls on the wall over here on the left-hand side. So he had apparently no apron on either, and the x-rays were coming from behind the patient. So you can see where that was, unfortunately, it resulted in some pretty high doses of radiation to his blood. Leukemia also was prevalent amongst patients with ankylosing spondylitis who were treated with radiation therapy, and they found that they had that linear non-threshold dose-response relationship. As far as the patients with thyroid cancer or thyroid nodules were all children. They were found to be in, two of them were series of testing that were conducted in Ann Arbor, Michigan and Rochester, New York, where they decided that if their child's thymus gland was enlarged, they would irradiate their neck. And when they did so with high doses of radiation, 20 years down the road, these people ended up with thyroid nodules or cancer. There was also some testing in the Marshall Islands in 1954, and on one of the atolls, they had children who were placed in a different atoll, but they wind shifted and went the other direction, and the vegetation and everything was completely covered with radiation fallout from the nuclear bomb testing. So as a result, several of the children in that island ingested a large amount of radioactive materials and developed thyroid cancer 20 years down the road as well. The dose-response relationship was confirmed to be linear non-threshold. Bone cancer was found in patients treated with radium or in radium watchdog painters. So bone absorbs calcium, and radium acts similarly in that it gets absorbed into the bone. So here's a picture of the radium girls, who they called them radium girls back in the day, where they would touch the tip of a brush with radium on it to paint dials on a clock, and the watch dials on the clock, and they all but one followed the instructions, and they all ended up dying from the effects of radiation. The one survivor just passed away a few years ago, did not follow the instructions, and left the employment of the company because she didn't like the taste of it. But a lot of the girls actually lost the sides of their faces, and they had huge goiters and things like that, and went to court over it. But most of them died before they ever got the court case went through. Other patients treated with radium salts were those with tuberculosis. There's the lady on the right-hand side who's in a TB sanitarium, and they also had other people who were treated with radium salts for conditions such as arthritis, and they resulted in having bone cancer. Breast cancer was found in people who were TB patients, tuberculosis patients in a sanitarium, just like the previous patients were with the arthritis, with the radium treatments. But in this case, they were inducing an artificial pneumothorax under fluoroscopy. And if they were prone, which was facing toward the X-ray tube, then they found that there was a higher incidence in breast cancer in those patients. And they were also patients treated for acute postpartum mastitis. They had inflammation of the breast postpartum, and they irradiated them to reduce the inflammation, and they developed breast cancer in 20 years. And finally, A-bomb survivors had a high incidence of breast cancer too. Breast cancer also was confirmed to be a linear non-threshold dose-response relationship. Lung cancer was found to be in miners. Bohemian Plinchman miners and American uranium miners were found in both groups. And the half-life of uranium is 10 to the 9th power in years, so it's 1 billion years. The decay product is radon. They inhaled the radon, and it broke down to alpha particles and damaged their lungs, causing lung cancer. And the other cancer was liver cancer. This was one in which we had close to home because it was used in angiography back in the 40s. So they used thorotrast, which was thorium dioxide, and it unfortunately is radioactive. And those individuals, when they injected the thorium dioxide into their veins for angiography studies, the particles were distributed into the spleen and liver, and they developed liver cancer. People who later developed skin cancer usually began with the development of radiodermatitis first, which is an inflammation of the skin due to radiation. This is the odd type of cancer that does not follow the rules, and that it's a dose-response relationship of nonlinear threshold. The latent period for this also is only 5 to 10 years, a shorter period than the 20 standard years for most cancers. Irradiation in utero occurred within population groups such as A-bomb survivors. So the children, they were carrying children while they were pregnant when the bombs were set off, and unfortunately the embryos and fetus were affected. So that if the woman was pregnant and she was within the first 2 weeks of the pregnancy, what they found with this group and with other groups was that they would have what's called prenatal death. And that's a spontaneous abortion where the child is aborted automatically. It's pretty rare, but only confirmed after high doses. It was observed, however, in another group, and that would be radiotherapy patients, women who didn't realize they were pregnant, but they had radiation therapy. And so they had a miscarriage in the first 2 weeks of the pregnancy. The result is all or nothing and considered to be of least concern because it's such an early stage of the pregnancy and happens so common anyway that during that stage that many women don't even realize that they're pregnant. In the 2nd to 10th week of a pregnancy are when the skeletal and organ abnormalities can transpire. They can also be found to have congenital abnormalities in the central nervous system if the pregnancy is carried to term. There can also be if the congenital anomalies are too severe, there can be neonatal death, which is being stillborn, born with them passed away. Now, as far as the thyroid gland, it is fully developed in a child, in an embryo at the 10th week. So if a mother needs to be administered radioiodine, it should be done in trace doses, which means small doses, and only before the 10th week, so that the iodine does not transfer over to the child or the embryo at all during the pregnancy. So radiation in utero was studied as far as the malignancy of greatest concern was leukemia, but the most radiosensitive period was the first trimester. Taking a look at the chart, we looked at what are called relative risk, and that's when you take the observed cases divided by the number of cases that you expect to see, and there's a greater risk that was found in the first trimester compared to the 2nd or the 3rd. So the first trimester is of greatest concern, and that's when the child is an embryo. Malignancy of greatest concern, as I already mentioned, is leukemia. So summary of effects after 100 milligram in utero were found to be at 0 to 2nd week would be spontaneous abortion, which is called prenatal death. Prenatal death. 2nd to 10th week were congenital abnormalities that we talked about. 0 to 9 months would be malignant disease, and I skipped one there, but there were some other items that were found too. Last but not least are genetic effects. There's 2 scientists that had conducted experiments. One was with fruit flies, which is this first one, and they found that there was a linear non-threshold dose-response relationship. It was found that these fruit flies reproduced fast enough to where they could see the genetic mutations, but there wasn't a difference in the quality, meaning the type of effect or how bad it was or severe it was, just about the amount of mutations that were occurring within the fruit flies. Another experiment conducted was by a man with the last name Russell. He had a mouse project that he found that some mice had a capability to repair the genetic damage, and he also confirmed the linear non-threshold dose-response relationship. Some conclusions that these scientists had from their studies were that the radiation-induced mutations are usually harmful, that any dose can cause some genetic risk, that's why they're referred to as non-threshold, that the frequency is directly proportional to the dose, so that's a linear effect, and it really depends upon protraction and fractionation. We talked about those earlier in this lesson. And then for most pre-reproductive life, the woman is less sensitive to than the man to the genetic effects of radiation. They also found that most radiation-induced mutations are recessive. That means that they have to be in both the male and the female to produce the trait in the offspring. Therefore, mutations may not be expressed for many generations. And finally, frequency of genetic mutations is extremely low. So it's even difficult to see when they actually transpire. So in a nutshell, we've talked about human biology, fundamental principles of radiobiology, molecular and cellular effects, and finally, deterministic and stochastic effects. Have a great day.